Imagine you're playing this really high-stakes game of chess. You've got your opponent completely cornered, right? You move your queen into position. You are exactly one move away from checkmate. And right as you're about to claim victory, your opponent just reaches out, changes all the pieces to flat disks, and declares you're now playing checkers.
Which means, every single brilliant chess strategy you just spent the last hour setting up is, well, it's now entirely useless.
Exactly. It is incredibly frustrating. But it's also the perfect visualization for what we are looking at today.
Welcome to the Deep Dive. Today we're exploring a groundbreaking paper from Nature Communications that exposes how cancer cells pull this exact trick.
Yeah, it's a biological process called drug-induced dedifferentiation.
Right. And the mission for this deep dive is to unpack the cancer cell's entire molecular escape route. I mean, from the exact second a drug hits it to the genetic switches it flips to survive. And more importantly, how we might actually stop them from changing the rules of the game. So to set the baseline here, the main subject of this study is BRAF mutant melanoma. It's a very aggressive form of skin cancer. Usually, the gold standard treatment is targeted therapy, specifically drugs like vemurafenib.
Okay, vemurafenib.
Right. And the thing you really have to understand is that initially, these drugs are practically magic. I mean, they work incredibly well at targeting and killing the cancer cells.
Yeah, but the operative word there is initially, right, because the terrifying reality is that often the tumor cells don't all die. Instead, they morph.
They do. They transform into this primitive stem-like state known as a drug-tolerant persister state, or DTP.
And this is where the paper really upends how we think about oncology, because the most mind-bending part of this persister state isn't just that the cells change the physical makeup, it's that the state is entirely reversible.
Yeah, that's the kicker. If you remove the targeted therapy, those highly resilient persister cells actually revert right back to their original vulnerable melanoma state.
Let's just pause on that for a second, because if you're listening and thinking about how cancer normally works, well, that shouldn't make sense. We are taught that cancer is a disease of genetic mutation, right? Like a permanent corruption of the DNA.
Exactly.
But if the cancer can just flip back and forth between vulnerable and invincible, based on whether the drug is floating around in the bloodstream, I mean, it means the underlying DNA sequence isn't changing at all.
Precisely. It is not a permanent mutation. It's an adaptive epigenetic change. The cancer is simply choosing to read different chapters of its existing DNA rulebook.
Wow.
And to prove this wasn't just some quirk of cells growing in a petri dish, the researchers looked at actual melanoma patients. The study highlights patient 1 and patient 2. Both initially responded beautifully to BRAF inhibitors, but unfortunately they relapsed.
And when they biopsied those relapsed tumors, what did they actually look like under a microscope?
Well, they found incredible spatial heterogeneity. It wasn't that the entire tumor uniformly decided to morph. It looked more like a patchwork quilt.
A patchwork?
Yeah. In the specific patches of the tumor that were successfully resisting the drug, the cells had completely lost their original identity. Normally, melanoma cells express a specific master protein called SOX10.
Okay, SOX10.
Right. SOX10 is essentially a name tag that says, I am a mature melanocyte. But in these resistant patches, the SOX10 name tag was completely gone. And they gained new markers, like KDM5B.
It's like the cancer cells just ripped off their uniforms so the drugs couldn't recognize them anymore.
Exactly. And they saw the exact same thing when they put human melanoma cells, specifically the M397 cell line, into mouse models.
Okay.
The physical transformation was completely dose-dependent. The mice, given the highest doses of the drug over the longest periods of time, showed the most extreme changes.
Makes sense.
Their tumors had a profound loss of that SOX10 identity marker, and they elevated N-coherent, which is a marker for a highly resilient primitive mesenchymal cell type.
So the cancer cell is essentially hitting a biological undo button on its own development?
Basically, yeah.
Let's use an analogy here. Say you have a highly specialized adult, like a renowned heart surgeon. Suddenly, the hospital they're working in catches fire. Instead of trying to use their, highly specialized surgical skills to survive the fire, which would be useless, they somehow instantly revert back into a resourceful, generalized toddler.
That's a great way to picture it.
Right, because a toddler has no idea how to perform heart surgery, but they are incredibly adaptable. They can crawl through small spaces and survive in environments the specialized adult just can't handle.
And crucially, the drug is a sniper that has been trained exclusively to look for a heart surgeon. When the drug scans the room and only sees a toddler, it assumes the target is gone and sails right past it. By shedding those specialized traits, the cell becomes functionally invisible to the therapy.
Okay, let's do a quick recap for you listening to make sure we're all on the same page. We're going to do these little summaries as we go. So, bullet point 1. Targeted drugs attack the cancer, but the cancer evades death by temporarily devolving into a primitive drug-tolerant state. And bullet point 2. This happens in both lab models and actual patients, which brings up our first big question for you to think about. If this process is completely reversible just by taking the drug away, it means the cancer isn't mutating its DNA. So what is it actually changing?
And when a cell does this, it is frantically flipping thousands of genetic switches all at once. The biological noise is just deafening.
So how do you even track that?
Well, to find the actual signal in all that noise, the researchers had to borrow a highly advanced algorithm from physics called surprisal analysis.
Surprisal analysis, which sounds like a magic trick, honestly.
It does, but it's actually an information theoretic math approach. It's used to understand systems that are completely out of equilibrium. It's designed to find the underlying forces driving the chaos.
Okay, so they feed the genetic data into this algorithm, and what happens?
By running the data through this algorithm, they boil those thousands of chaotic fluctuations down into just 2 distinct sequential transcriptional waves. They call them M-early and M-late.
M-early and M-late, got it.
The M-early wave hits incredibly fast. It's fully active within just 3 days of the drug touching the cell. It involves histone modifications, and it basically acts as the brakes. It forces the cell into cell cycle arrest.
Which makes sense. If you are under attack, you stop building new rooms on the house and focus on putting out the fire. And then comes the M-late wave, which I'm assuming is a slower burn.
Yes, M-late takes weeks to fully activate.
Yeah.
It actually inverts around day 21. And this is the wave that actually drives the physical change into that neural crest or mesenchymal toddler state.
Right.
But the most critical finding from analyzing these 2 waves is a concept called hysteresis. Hysteresis means that the path the cell takes to enter this drug-tolerant state is not the same path it takes to get out.
Oh, wow. Okay, this reminds me of a memory foam mattress.
How so?
Well, when you press your hand heavily into memory foam, it deforms instantly. But when you lift your hand up, it doesn't just snap immediately back into a flat surface, right? And it holds a memory of the pressure. It takes time for it to slowly rise back up.
Yes. And that biological memory is physically recorded in the cell structure. The researchers found that even when 2 cancer cells look entirely identical on the outside, like they both look like fully transformed drug-tolerant cells in their M-late profile, their internal chromatin holds a secret.
The early wave profile.
Exactly. Their M-early chromatin profile holds a memory of whether the drug was just applied or if it was just removed. It's a one-way hysteretic loop, not a straight 2-way street.
Okay, time for another quick recap. Bullet 1. Cancer's escape relies on 2 waves, an early preparation wave, M-early, and a late physical transformation wave, M-late.
Spot on.
Bullet 2. These waves move in a one-way loop called hysteresis rather than a straight line. But that leaves a massive question for you listening. Since M-early acts as the trigger, what exactly is the drug doing to the cell in those first 3 days to kickstart this wave?
And that brings us to what I think is the most fascinating paradox in the entire paper. It's the irony of the cure.
The irony of the cure. I love that phrasing.
When you give a patient a targeted inhibitor, you are successfully shutting down the cancer's main driver gene, BRAF. But as an unintended side effect, the drug severely disrupts the cancer cell's natural antioxidant defenses.
Wait, really? So the protective shields just drop?
Entirely. Critical protective genes like NFE, 2L2, and TXN are immediately downregulated. And because the cell loses its defense system, it experiences a massive toxic buildup of reactive oxygen species, or ROS.
Okay, ROS.
You can think of ROS as highly corrosive cellular exhaust. It causes immense oxidative stress.
Think about what that means for a second. The very drug that is designed to quietly assassinate the cancer cell is inadvertently filling the room with smoke. The ROS buildup is the exact signal the cancer needs to realize it's under lethal attack. It's literally providing the cancer with its own warning system.
It is. And that smoke triggers a very specific mechanical domino effect. Step 1. The massive influx of ROS inactivates specific phosphatases, basically housekeeping enzymes called PP2A, PP2B, and PP2C.
Okay, so the smoke knocks out the housekeepers.
Right. Step 2. With those knocked out, a very specific repressor protein in the cell called NFKBIE, let's just call it the bouncer, gets phosphorylated and completely degraded by the cell.
So the drug causes smoke, the smoke knocks out the housekeepers, and the bouncer is destroyed. What was the bouncer guarding?
That's step 3. The bouncer's only job in a healthy melanoma cell is to hold a crucial transcription factor called RelA hostage outside the nucleus. RelA is part of the NFKB family. As long as the bouncer is alive, RelA is not allowed near the DNA.
But the toxic exhaust just killed the bouncer.
Exactly. So step 4. With the bouncer gone, RelA floods directly into the cell's nucleus, and its arrival officially kickstarts that rapid M-early wave.
Okay, let's give the listeners a mnemonic device to remember this dense cascade because it's a lot. Let's call it the RRES escape plan.
RRES, I like it.
So RRS builds up. The second RelA is unleashed, E, epigenetics change, and S, SOX10 is silenced, RRES.
That's a perfect way to remember it.
Awesome. Let's do a mini recap. Bullet 1. The drug ironically causes oxidative stress, or ROS, bullet 2. That ROS degrades the cellular bouncer, allowing RelA to rush into the nucleus and start the early wave.
Exactly.
Which brings us to our next listener question. Once RelA gets into the nucleus, it's just a transcription factor, right? So how does it physically shut down the cancer cell's original identity?
Right, because RelA cannot mutate the DNA. But it doesn't need to, because it doesn't work alone. Once it breaches the nucleus, RelA recruits 2 specific epigenetic erasers called KDM5B and HDAC1.
Okay, let's go back to an analogy to make this concrete. If you're picturing this, imagine the cancer cell's entire DNA genome is a massive, sprawling library containing thousands of books.
Okay.
RelA is a rogue librarian who desperately wants to hide certain books from being read. But he can't burn the books. The DNA can't be destroyed. So instead, RelA calls in his friends, KDM5B and HDAC1, the epigenetic erasers. They are basically heavy-duty padlocks.
I love this.
They don't destroy anything. They just march into the library and permanently padlock the doors to specific aisles, so those books can literally never be checked out.
And the specific aisle they target is the promoter region of the SOX10 gene. Remember, SOX10 is the master gene that makes the cell melanocyte. When RelA and these padlock enzymes arrive at SOX10, they erase the specific activation marks, specifically H3K4ME3 and H3K27AC on the histones.
So without those activation marks, the chromatin just coils up tight.
Tightly coiled and plunged into darkness. The SOX10 gene can no longer be transcribed. And without SOX10, the cell completely loses its melanocyte identity, upregulates TGF-beta signaling, and becomes a drug-tolerant mesenchymal cell.
What's incredible about this paper is they didn't just passively observe this library getting locked down, they proved it mechanically.
Yeah, they did. They used CRISPR to genetically knock out NFKBIE, the bouncer, in the lab. And instantly, without even giving the cells a drop of the cancer drug, RelA flooded the nucleus, locked down SOX10, and the cells developed drug tolerance.
Wow, and they proved it from the other direction too, right?
They did. They used a drug called JSH23, which blocks RelA from entering the nucleus. When they treated the cells with that, the chromatin stayed open, SOX10 stayed active, and the cancer cells just died when hit with the standard therapy.
All right, quick recap time. Bullet 1. RelA acts as a foreman, bringing in KDM5B and HDAC1 to epigenetically lock away the SOX10 gene. Bullet 2. This strips the cell of its original identity, which leads to a really important listener question. This is brilliant for melanoma, but cancer is wildly diverse. Could lung or colon cancer really be using this exact same escape hatch?
You would naturally assume the answer is no.
Right.
Right, because they originate from entirely different tissues. But the researchers tested a cell line called HCC827-, which is an EGFR mutant lung cancer treated with erlotinib.
Okay.
And they tested HT29, which is a BRAF mutant colon cancer treated with dabrafenib and cetuximab.
And what happened?
Both lines use the exact same ROS to RelA pathway to become drug tolerant.
That is staggering. It means this isn't just some quirky trick isolated to skin cancer. It's a universal biological escape route.
It really is.
Yeah.
But the paper goes even further and introduces a concept that I think is, well, it's mind-blowing. Pre-encoded plasticity.
Pre-encoded plasticity. Break that down for us.
It means a cancer cell's ability to shape-shift into this toddler state isn't random. It is predetermined by how loose or accessible its chromatin is before the drug ever even touches it. Highly plastic cells essentially have a hair-trigger chromatin state.
Think about that. The cells that easily escape the drug already have their library doors well-oiled and slightly ajar, just waiting for the RelA librarian to come slam them shut.
Exactly, which leads to a massive new therapeutic arsenal. This completely changes how we treat patients. Instead of just giving the targeted therapy, which triggers the escape route, we can co-administer drugs that block the escape.
Like we can set traps.
Exactly. You could give NAC, which is an antioxidant, to neutralize the ROS so the alarm never rings.
Or give JSH23 to stop RelA at the door.
Or give CPI 455 or quizanostat to stop the epigenetic erasers KDM5B and HDAC1. If you take away the padlocks, they can't lock the doors. And the paper shows these combinations successfully wiped out the persister cells without massive toxicity.
Time for our final recap. Bullet 1. This RelA epigenetic mechanism is a universal cancer escape hatch. Bullet 2. We can proactively trap the cancer by giving targeted therapies alongside epigenetic or ROS blocking drugs.
Exactly.
And here's the final listener question to consider. If cellular plasticity is pre-encoded in the chromatin, could we one day biopsy a tumor and predict exactly how it will try to evolve before we even treat it?
We could be 2 steps ahead of the cancer before the chess game even begins.
It's just a profound thought. Cancer isn't just some brute force genetic mutation machine. It is an incredibly elegant, adaptable system. It survives by exploiting the very biological flexibility that once allowed a single embryo to become a complex human.
And that leaves us with a final thought to mull over. We spent decades trying to design stronger poisons to kill cancer. But if cancer uses the stress of our drugs to trigger its own survival program, maybe the future of oncology isn't about poisoning the cell harder. What if the ultimate cure relies on keeping the cancer cell relaxed? Yeah, locking it into its current identity so it simply forgets how to shapeshift.
By keeping the cell calm, we ensure it never realizes it's under attack. So, the next time you picture that chess match against cancer, remember, the goal isn't just to aggressively capture the king. The real goal is to make sure your opponent never even realizes they have the option to change the pieces to checkers. Thanks for joining us on this deep dive.